Greetings and welcome to the Tectonic TX45 Phase 1B Interim Data Update Call. At this time, all participants are in a listen-only mode. A question-and-answer session will follow the formal presentation. If anyone should require operator assistance during the call, please press *0 on your telephone keypad. It is now my pleasure to introduce your host, Ms. 𝗖𝗢𝗡𝗘𝗖𝗧, Chief Executive Officer. Thank you. You may begin.
Thank you. Good morning, and thank you for joining us. Let's go to the next slide, please. Before we begin, on this slide, you will see our standard disclaimer language. Statements in this presentation and on this call may be forward-looking, including our plans, objectives, and expectations for our TX45 program. And with that, let's go to the next slide and get started. At Tectonic, we're leading the way in the discovery and development of GPCR-targeted biologics, the development of which has been a real challenge in the field. The company was founded in 2019 by Tim Springer and Andrew Kruse. We went public via reverse merger in June 2024, along with a $131 million private placement. We have an unusually experienced and successful team. Between us, we've led teams resulting in over 20 first approvals across therapeutic areas, enabling us to choose what we think are the highest POS targets regardless of therapeutic area. Our focus today will be exclusively on our long-acting relaxin program, for which we are now in Phase 2 with highly supportive Phase 1B data that I'll be reviewing today. The initial indication targets Group 2 pulmonary hypertension, which is associated with preserved ejection fraction heart failure, or I'll be referring to it as PH-HFpEF, with an emphasis on the subpopulation of patients with combined pre- and post-capillary pulmonary hypertension. Our Phase 1A study in normal healthy volunteers confirmed both the safety of the drug and that we have a potential best-in-class therapeutic. The Phase 1B results that we are sharing today met and/or exceeded our targets, and we believe that they increase the probability of success of our Phase 2 study. Go to the next slide. Oh, no, please go back. I shouldn't have said that. I apologize. To be clear, we considered multiple indications for TX45, including the broader HFpEF population, but we chose our initial indication to be PH-HFpEF, enriched for CTC, because we believe the multimodal action of relaxin really matches the pathophysiology of the disease, and I think our Phase 1B data demonstrate this. Furthermore, PH-HFpEF is a very large population with over 1 million patients in the U.S. alone, high 5-year mortality, and no approved therapies, making it what we believe to be a multi-billion-dollar opportunity. We are not alone, as AZ is exploring both PH-HFpEF and PH-HFrEF. That's pulmonary hypertension in the setting of reduced ejection fraction heart failure. We recently expanded our Phase 1B study into PH-HFrEF with data from that population in second half of this year. And going into PH-HFrEF could almost double the patient population. Go to the next slide. There are 2 subtypes of pulmonary hypertension associated with heart failure: IPCPH, isolated post-capillary pulmonary hypertension, or CPCPH, combined pre- and post-capillary pulmonary hypertension. IPCPH is where you have elevated pressures in the left side of the heart due to heart failure that then backflow into the pulmonary circulation, leading to elevated pulmonary pressures. The pulmonary vasculature is normal. However, patients who have IPCPH are at risk after years of the disease of developing CPCPH, where they have both that post-capillary component, i.e. the backflow, but in addition, they have a precapillary component. What does that mean? It means they get muscularization of the arteries, their pulmonary arteries, with a narrowing of the lumen. It almost looks like they're in their pulmonary vasculature, they have early pulmonary arterial hypertension. And as blood is pushed through a smaller pipe, you get an increase in pulmonary vascular resistance, or PVR, which is how you differentiate these patients from the IPC-PH patients who have a normal pulmonary vascular resistance. Importantly, in our phase 2 study, we're going to enrich for CPC-PH patients who we believe have the greatest potential opportunity of benefit from relaxin. In terms of the epidemiology, which is shown on the left side, the broader population of preserved ejection fraction heart failure HFpEF affects about 4 million patients in the U.S., and about 1.4 million of them are New York Heart Association Class 2 and Class 3 and have elevations in their pulmonary artery pressure and therefore have PH-HFpEF. Of these, depending on how you define CPC-PH, and in the past it was defined as a PVR greater than 3 units. More recently, it's defined as a PVR greater than 2 units. If you define it as a PVR greater than 2 units, there are 700,000 CPC-PH patients and 700,000 IPC-PH patients. Of note, adding group 2 PH-HFrEF would close to double the population, as there are about 1.1 million patients with PH-HFrEF in the U.S., with a similar proportion of CPC and IPC patients. Go to the next slide, please. Relaxin has multiple different effects, all of which we think should be beneficial in the treatment of group 2 pulmonary hypertension. In the phase 1b study that I'm going to show you data from, we should be measuring the vasodilatory effects in both the pulmonary and the systemic circulation, which should decrease afterload on the right and the left ventricles, making it easier for the heart to pump blood out, as well as what's called its lusitropic effects, the ability of relaxin to actively relax the left ventricle, which should improve the diastolic dysfunction of the left side of the heart in HFpEF and enable it to better fill with blood during diastole. This might be especially important in CPC-PH, as if you can reduce both the pulmonary vascular resistance, which should then send more blood into the left side of the heart. And if you enable the heart to relax to be able to accept that blood, you should be able to then get an increase in cardiac output and an increase in stroke volume, which should increase the exercise capacity of these patients. We also think relaxin has an antifibrotic and remodeling effect. We do not think we will be seeing those effects in this phase 1b study, but hopefully those effects will become apparent in our phase 2 6-month study. Next slide, please. TX45 is a human relaxin-2 Fc fusion protein with a potential best-in-class half-life of 2 to 3 weeks, which we are in phase 2 now, studying both monthly and every other week dosing. Our phase 1b hemodynamic study is ongoing. Enrollment and dosing in the PH-HFpEF Part A cohort has been completed with an N of 19, and the data we'll be presenting today came from the first and only interim analysis we've done of this study. In this interim analysis, efficacy data is available for 16 of the 19 patients enrolled. Why, you might ask, why are you presenting this data why not just wait for the full data set? We thought the data was compelling and clear, and although not included in the analysis because their full data wasn't available, at the time this analysis was conducted, we have seen the hemodynamic data on those last 3 patients, and their data is completely in line with the current analysis, so I do not expect any significant changes in the final analysis that we will present at a medical conference. Also importantly, the last 3 patients who are not included in this analysis all have isolated post-capillary pulmonary hypertension, IPCPH. So the CPCPH cohort is final. Additionally, safety is available on 16 patients, of whom 15 have completed the full 43-day safety follow-up. We look forward to sharing these data with our phase 2 investigators as we think it will drive momentum in our program. We hope to start enrollment in the PH-HFrEF portion of the study, Part B, with data anticipated in the second half of this year. And our phase 2 study is currently enrolling with data expected in 2026. Go to the next slide. In the phase 1B study, patients get admitted to the unit and a right heart cath is placed. Hemodynamic measurements are taken at 30 and 60 minutes prior to dosing, and the average of these is the baseline for each patient. They then receive an infusion of 0.3, 1, or 3 milligrams per kilogram of TX45, and hemodynamics are measured at hours 1, 2, 3, 4, 6, 7, and 8. And we pre-specified that the change from baseline would be determined as the average of these 7 time points. It's important to note that the study was designed to evaluate hemodynamic data pooled across all 3 of these doses. After IV administration, all dose levels that are studied here result in exposures during the 8-hour assessment period, which are in the predicted efficacious range. So we didn't expect to see a dose effect. The only reason, in fact, that we did dose escalation was for safety, since this study was started when we were still doing dose escalation in our normal healthy volunteer phase 1A study. Go to the next slide, please. We strongly believe that for a drug to work in patients with PH-HFpEF, and especially in the subgroup of patients with CPCPH, the drug needs to do 2 things. It needs to improve the function of the left side of the heart and improve the pulmonary hemodynamics. We measured a variety of hemodynamic measurements, and I thought it would be useful just to take you through those before we get into the data. You're going to hear me talk a lot about pulmonary capillary wedge pressure, or wedge pressure, which measures left atrial pressure and is a key marker of pressures on the left side of the heart and left ventricular function. PVR, pulmonary vascular resistance, measures the resistance to blood flow in the pulmonary arteries. And the equation for this is mean pulmonary artery pressure minus pulmonary capillary wedge pressure divided by cardiac output. Total pulmonary resistance, TPR, measures the afterload, or the pressure that the right ventricle needs to pump against. The equation for this is mean pulmonary artery pressure divided by cardiac output. Cardiac output is the amount of blood the heart pumps out, and the equation is stroke volume times heart rate. And stroke volume, of course, is the amount of blood ejected from the left heart per beat. Go to the next slide, please. When we set up the phase 1B study, the team set very clear goals for what we would consider a success. And over the past 18 months, we have been communicating these to investors. I can now tell you we met and/or exceeded all of the success bars we laid out. As I just said on the last slide, we strongly believe that we needed to have evidence of both an improvement in left ventricular function and the pulmonary vascular dysfunction seen in these patients. Therefore, it's important to demonstrate that TX45 lowers wedge pressure on the order of 15 to 20%, because pulmonary capillary wedge pressure, which is, as I said, a measure of left heart function, is the key hemodynamic marker that correlates at rest with exercise capacity in HFpEF patients. In addition to lowering wedge, in the CPC HFpEF patients, we are also looking to lower PVR on the order of 15 to 20%. Why not include the overall cohort and the IPC-PH patients? Well, IPC-PH patients have a normal PVR, and so we don't think they'll be possible to see a reduction in these groups because of a floor effect. We have no successful therapies in PH-HFpEF, so there's very little data to tell us exactly how much lowering of PVR will translate into an improvement in exercise capacity for this population. However, we know there is an important link demonstrated between PVR and 6-minute walk test, as has been demonstrated by many successful trials in pulmonary arterial hypertension, or PAH. Across multiple mechanisms in PAH, therapies have demonstrated lowering of PVR on the order of 15 to 20% is associated with clinically significant improvement in 6-minute walk test in patients with PAH. And studies have also demonstrated a correlation between PVR and pulmonary capillary wedge, as well as and 6-minute walk test in Group 2 PH. In PH-HFpEF, lowering of PVR alone is not enough to provide efficacy, as has been demonstrated in many failed trials looking to repurpose PAH therapies for the PH-HFpEF population. Lastly, in this study, we're exploring the impact of TX45 on total pulmonary resistance. This endpoint would be relevant both for the IPC and CPC PH patients. A reduction in TPR would be an indication that we've reduced right ventricular afterload. In other words, we've reduced the pressure that the right ventricle is pumping against, which should be important in the prevention of right heart failure, which is often what kills these patients. The limitation of this measurement is that we're not aware of extensive literature linking magnitude of change in this endpoint to changes in 6-minute walk distance. Lastly, we did not set a bar for cardiac output in this study, as cardiac output is normal in patients with PH-HFpEF at rest. And while we expected it to increase during exercise, we were not certain we would see this at rest, which is when we are doing our hemodynamic measurements. But we did say if we saw it at rest, it would provide further confidence that cardiac output would also increase with exercise and therefore lead to an improvement in exercise capacity and 6-minute walk test. Next slide. Baseline characteristics are generally in line with what you might expect for this patient population. Typically, a third of the patients are obese, which goes along with a mean BMI skewing high at 28. Their mean creatinine is normal. 81% of the patients in this study have hypertension. The majority have atrial fibrillation. A third have diabetes mellitus, and over 60% have coronary artery disease. And the population was pretty evenly distributed between New York Heart Association Class 2 and Class 3. The patients were on multiple cardiovascular therapies, including 50% on ACEs and ARBs, over 80% on an MRA, close to 40% on SGLT2s, 70% on loop diuretics, 75% on beta blockers, and a third of the patients on digoxin. Next slide, please. Mean systolic blood pressure was just slightly over the normal range. Pulmonary artery pressure of above 26 is what you would expect. The normal for pulmonary artery pressure is 20. Pulmonary capillary wedge pressure of 12 is normal, and we had a mean of 16.7, consistent with heart failure. And on the right, you can see if we define CpcPH with the old criteria of a PVR of greater than 3, then 11 patients had IpcPH and 5 patients had CpcPH. Using the new criteria of a PVR greater than 2, we had 7 patients with IpcPH and 9 with CpcH. And for completeness, you'll see us present the data in both ways. Go to the next slide. The primary endpoint for the phase 1b study was safety. TX45 was well tolerated. There were no SAEs, severe adverse events, discontinuations, or infusion-related reactions. The table presented here summarizes the treatment-emergent adverse experiences. There were no clinically significant changes in vital signs, EKGs, or lab values. I will be talking about blood pressure in 2 slides. The most common TEAE was fatigue, which was mild to moderate and transient, occurring on the first day of dosing. In fact, we think it's procedure-related because it was the end of that very long day in the ICU where they had a right heart cath in, and by the next morning, when drug exposures were still high, the patients were asymptomatic. Next slide. And finally, a summary of our data. Our phase 1b acute hemodynamic data, again, I'll repeat, met or exceeded all of our stated goals. This slide has the mean change from baseline, that's what CSB means, along with 95% confidence intervals. And to just remind you, if the 95% confidence interval doesn't include 0, that means that the value is statistically significant with a p-value of 0.05. Because we didn't have hierarchical testing, I would have to say they were of nominal statistical significance. But as you can see on this slide, every single one of these 95% confidence intervals excludes 0. We had a meaningful decrease in wedge pressure of almost 3 millimeters of mercury and 18% in the total population, and a mean decrease of PVR of 32% in patients with a baseline PVR of greater than 2, and a mean reduction of 35.5% in patients who were defined as having CpcPH with a PVR of greater or equal to 3. While we didn't set a bar for cardiac output increase, because as I said, patients with HFpEF typically have normal cardiac output at rest, we and our advisors are very encouraged to see an improvement in cardiac output by over 17%, and a corresponding increase in stroke volume of 14%, suggesting that the increase in cardiac output is mainly driven by the increase in stroke volume rather than an increase in heart rate. We also saw significant improvements of 26% in total pulmonary resistance and a 16% reduction in the mean pulmonary artery pressure. Next slide. IV administration of TX45 was associated with a transient asymptomatic 5 to 12 millimeter mean decrease in blood pressure on the first day of dosing, which resolved at subsequent visits. Increases in heart rate were in the single digits. These were not clinically significant, and we anticipate that with sub-Q dosing, the first-day effects will be even smaller. Go to the next slide. As I go through the next few slides, I want to point out the row that says all subjects includes the entire patient population, both IPCPH and CPCPH, regardless of definition. And as I said before, as you can see in the overall population, we had an absolute decrease of 3 millimeters of mercury. Going into this, our advisors had told us on an absolute basis that a 3 millimeter change would be highly encouraging and is important. What you can see in the bottom is that steady state reduction in the wedge pressure isn't achieved until about 2 or 2.5 hours after dosing. And I think you'll see something quite similar in the rest of the hemodynamic endpoints. And just to remind you, we included all the data from hour 1 on. And so by including the data from hours 1 and 2 in our mean effect, we probably, if anything, have slightly underestimated the physiologic effects. And I will point out that the effects are maintained over the dosing interval. Go to the next slide. In the overall population, which included the IPCPC patients, we had about a 12% decrease in the overall population. It was really driven by the CPCPH patients. Basically, we saw no reduction in the IPCPH patients. Also, there was a very strong correlation, although I don't show it on this slide, between baseline PVR and the change in PVR with a Pearson's R ratio of 0.89, which had a p-value of less than 0.05. Next slide. TPR, which is the pulmonary pressure over cardiac output, and which represents, as I've said, pressure that the right heart sees, decreased in the whole population, suggesting that the stress on the right heart, which ultimately leads to right heart failure, was reduced in the overall population, both in the IPC population and the CPC population. And we also saw a 16% improvement in mean pulmonary artery pressure. We were encouraged, as I said before, to have seen an increase in cardiac output of 16%, and we think it bodes well for the potential to increase cardiac output with exercise in future studies. It is known that the ability to augment cardiac output is essential to improve exercise tolerance. Go to the next slide. This is just, as I've shown you on other ones, to show you that both TPR and cardiac output were increased both in the overall population and in the CPC-PH population. Next slide. 6-minute walk test is the approvable endpoint. So what everybody wants to know is, are there any data that ties the hemodynamic effects that you've seen to 6-minute walk test? And unfortunately, unlike PAH, there's just limited literature to guide us, but the literature that does exist is quite reassuring. Decreasing wedge pressure in these patients alone is expected to improve exercise capacity to some extent. In patients with preserved ejection fraction heart failure, SGLT2 inhibitors were shown in one study to result in a decrease of pulmonary capillary wedge pressure of about 20%, and in a subsequent study in a similar patient population to increase 6-minute walk test by 20 meters, an improvement which is felt to be clinically significant in this population. However, based on our understanding of exercise physiology, improvement in pulmonary vascular resistance, in addition to improvement in wedge pressure, should further improve exercise capacity in patients with CP-CPH-HFpEF, because we know that in that patient population, what impairs their exercise capacity is both their PVR, which reduces the amount of blood they can send into the left ventricle, as well as the ability of the left ventricle to pump that blood out. In a study in CP-CPH patients who underwent an experimental surgical procedure called pulmonary artery denervation, they saw a treatment-adjusted decrease of about 19% in the wedge pressure, which was accompanied by a 30% decrease in PVR and resulted in an increase in 6-minute walk test of 70 meters. Now, I am not trying to claim that our expectation is that we will see a 70-meter increase in 6-minute walk test because these patients had very severe CP-CPH. What I am trying to claim from this is that you might get an either greater benefit in exercise capacity by reducing both pulmonary capillary wedge pressure and PVR, as the pathophysiology of this condition would suggest. Go to the next slide. In summary, we're encouraged by your interim phase 1b data, and we think these data should improve the probability that TX45 will be an efficacious therapy for the treatment of PH-HFpEF, and in particular in patients with CP-CPH. TX45 was safe and well tolerated. There were transient decreases in blood pressure over the first 24 hours, which resolved. We have clear evidence of improvement in both left heart function and pulmonary hemodynamics, and we believe both are important in this patient population. We have a differentiated profile from previously failed repurposed PAH therapies in that we improve wedge pressure, cardiac output, as well as PVR. Lastly, these data support our focus for our initial indication in PH-HFpEF with our enrichment strategy for patients with CP-CPH and an elevated PVR. Given these data, we have also decided to explore effects of TX45 in PH-HFrEF, that's patients with reduced ejection fraction, in this study as a Part B. Similar improvements in hemodynamics in the PH-HFrEF population may allow us to further expand into this population as well. Next slide. Looking forward, we are currently enrolling our phase 2 APEX 24-week study in patients with PH-HFpEF, which is highly enriched for patients with CP-CPH. In that study, after obtaining baseline hemodynamics via right heart cath, patients are randomized 1 to 3 weeks later in a 1-to-1-to-1 manner to receive subcutaneous dosing regimens of either placebo, 300 milligrams of TX45 every 2 weeks, or 300 milligrams of TX45 every 4 weeks in a double-blind, double-dummy manner. And after 24 weeks of treatment, patients will have a repeat hemodynamic assessment. Our primary endpoint is change from baseline in pulmonary vascular resistance, and a key secondary is change from baseline in pulmonary capillary wedge pressure. We will also be assessing whether TX45 has a treatment effect on 6-minute walk test and KCCQ. And we are aiming to have top-line data from this trial in 2026. Next slide. Just one slide reminding you how we selected doses for our phase 2. In chronic rat models of pulmonary hypertension, we found that an EC70 and a predicted human exposure of 2 micrograms per ml at trough was associated with maximal efficacy. Using repeated PK and renal blood flow in our phase 1 study, and renal blood flow was our pharmacodynamic measure, we created a data-rich PK/PD model, which is shown on the right. Doses of 300 Q2 and 300 milligrams Q4 provide maximal to near maximal agonism of RXFP1 throughout the entire dosing interval with trough exposures of 8.7 and 2.6 micrograms per ml. As you can see on the left, over the 2 to 4 week dosing interval, the 3 milligram per kilogram IV dose looks closest to the 300 milligram sub-Q dose, while the 0.3 milligram per kilogram dose has significantly less exposure over that time frame, but on day 1, as I said, is in the efficacious frame. All 3 doses, I'll just remind you, in our phase 1b study had exposures above the 2 microgram threshold throughout the 8-hour dosing interval. Last slide, please. We expect to get data, as I said, in our 1b cohort in PH-HFpEF in the second half of this year, and from our phase 2 study in 2026. I'll end by saying we're extremely encouraged by the results we've shown you today and for the potential benefit of TX45 to treat patients with this severe cardiopulmonary disease, which has no approved therapies. We are aware that other relaxin moieties have failed in different patient populations using different endpoints. The patient population and the hemodynamic endpoints are similar both in our phase 1b and our ongoing phase 2, where the gold standard right heart cath was used to measure hemodynamic changes. And we think the data that we showed you today strongly supports our phase 2 program. With that, thank you for listening. We'll open it up for question and answers.
Thank you. We will now be conducting a question and answer session. If you would like to ask a question, please press star 1 on your telephone keypad. A confirmation tone will indicate your line is in the question queue. You may press star 2 to remove yourself from the queue. For participants using speaker equipment, it may be necessary to pick up your handset before pressing the star keys. One moment, please, while we poll for questions. Our first question comes from the line of Tyler Van Buren with TD Cowen. Please proceed with your question.
Hey guys, congratulations on the tremendous results in achieving clinical proof of concept for the relaxin agonist class. Again, this is very exciting data, but this is an 8-hour time point. So what gives you confidence that these clinical effects will be sustained over a longer time period with the subcutaneous formulation and ultimately translate to functional outcomes in phase 2 and phase 3? And second question is just curious to get your thoughts on the discontinuation of the Lilly program the other day. Why or why not do you think it has any read-through to TX45?
Tyler, thank you. Both excellent questions. And I think asking about the long-term effects was a critical question. I'm going to give you 3 things that I think are reassuring to me. The first is on a molecular level, you don't get desensitization of the RXFP1 receptor for relaxin with prolonged agonism. Some GPCRs can get desensitized with chronic agonism. The way that's done is they couple with beta-arrestin, and then the receptor gets internalized. RXFP1, the receptor for relaxin, does not couple with beta-arrestin, and therefore we know it doesn't get desensitized. Secondly, in the studies looking at renal blood flow, both ours and others, in renal blood flow, the drug relaxin increases renal blood flow, and it does that as part of its systemic vasodilatory effects. So when renal blood flow remains elevated, it's an indication that the effects are prolonged, And we saw in those studies that the increase in renal blood flow that was seen on day 2 was also seen out a month in the study, so prolonged. There was also a study many, many years ago done in patients with scleroderma who received a continuous IV dose of a short-acting relaxant for 6 months. And when that dose was abruptly stopped, they saw a deterioration in their kidney function, which tells you, and an increase in creatinine, which tells you there was a prolonged effect of the drug on their kidneys. And then lastly, we didn't talk about it today, but when I was in medical school, relaxin was called the hormone of pregnancy because it's markedly upregulated during pregnancy. And what does it do in the pregnant mom? It dilates her pulmonary and cardiovascular circulation in order to enable her to increase cardiac output to sustain the demands of the developing fetus. People aren't pregnant for 8 hours. They're pregnant for months. And that also provides me reassurance that these effects will be long-lasting. But I can't show you data in the patient population that we are studying. That is going to come from our phase 2 study. Now, you asked about Lilly. At a very simplistic level, I will say it was a different endpoint, it was a different population, it was a different molecule. I think the first 2 are probably the most important. In our phase 1B and in our phase 2, we're using a gold standard for measuring hemodynamics, measuring endpoints that we know have a strong correlation to 6-minute walk test. In the Lilly study, they used echocardiographic endpoints. Doing echo in a large multi-center study can be quite difficult. There's a lot of user variability. And they used a really interesting endpoint, left atrial reservoir strain, which to the best of our knowledge has never been used in a large multi-center therapeutic study. And it's not clear to us that it's dynamic and can actually be responsive to therapy. In fact, the one study we found where it was used to look at patients who had mitral valve replacement, their heart failure got better, but the endpoint did not get better. Now, we haven't seen the Lilly data, so I can't speak beyond that on the specifics, but the endpoints are different. Lastly, the patient population. As you heard from us, we considered the broader HFpEF population and thought there could be some efficacy in that population because of what I described of the fact that we think that particular the CpcPH patients, where we think both the reduction in the pulmonary vascular resistance and the improvement in left heart function should have additive or better effects on exercise. We really thought that would be the ideal patient population for this disease. And it may mean if this drug doesn't work extremely well in HFpEF, we might see less efficacy in the IPC-PH population. And that's why we've enriched for CpcPH. We didn't want to miss a strong signal in that patient population. And we've been saying that since the beginning of our program. Lilly had an active compound. We know that from the renal blood flow data they showed in normal healthy volunteers. I don't know the dosing that they chose for their study. So I think we just have to wait for more data come out from Lilly. I think our data stands on its own for our program, our patient population, our molecule. Any other questions?
Nope, that's great. Thank you very much.
Thank you. Our next question comes from the line of David Risinger with Leerink Partners. Please proceed with your question.
Thanks very much, and let me add my congrats on the phenomenal data as well. So my two questions are, first, 𝗖𝗓𝗓, could you provide a little bit more clarity on your statement that Phase 2 APEX is enriched for CpcPH? Do you envision enrolling the vast majority of patients with CpcPH? And then second, what are key considerations for the upcoming AstraZeneca Phase 2 trial readout in the second half of 2025? I think that this is also a situation where it's, I guess how you described for Lilly, different population, different endpoint, and different drug, but would love to have you help us better understand how you're thinking about that trial readout and implications if it's positive or negative.
Okay. So yes, the overwhelming majority of patients in our Phase 2 will have CpcPH. I haven't given more specifics to that, and I have also said we're going to look at our Phase 1b data, and we may make some modifications to that study based on that. Right now, we feel very comfortable with that. We want to review these data with our steering committee. Once we've made any decisions about whether we are or not going to make any small changes to the protocol, we'll be ready to share more specifically how we designed that study. But I can assure you it was highly enriched for CpcPH patients. AZ, like us, I think really thought that the greatest benefit would be in PH-HFpEF. They just published a design study, and while they're not specifically enriching for CPC by enriching for patients with a high PVR, they have significantly enriched it for patients with pulmonary artery pressures above 25. And typically the patients that have very high pulmonary artery pressures above 25, many of those will have CpcPH. So they have taken a different design to enrich. I think we'll want to see when they present the data in CPC versus IPC, and understanding that will be important in interpreting the data. They also have HFpEF, preserved ejection fraction, and reduced ejection fraction heart failure. We think the drug has the potential to work in both populations. We'll be looking to our Phase 1b data to get a sense of whether it seems to work better in one or the other population, and obviously understanding that will also be important when the AZ data comes out to understand that. I think also understanding how AZ, what doses they chose, how they chose their doses. They didn't do renal blood flow in their Phase 1 study, but they do have a renal blood flow study ongoing at this time.
Great. Thank you very much.
Thank you. Our next question comes from the line of Yasmeen Rahimi with Piper Sandler. Please proceed with your question.
Good morning, team. Congrats on the outstanding data. I guess the question is, I think you guys recently, beginning of the year, enrolled a Part B study in HFrEF, which I think now you got it to have top-line data in the back half of the year. So would love to hear if there's anything we can infer from this data, even though obviously HFrEF is a very different population than HFpEF, that could just sort of allow us to think about the POS of success there. So if you could allude that. And then maybe one other question that we've been getting is maybe just around thoughts around, an editorial around efficacy of long-acting relaxin molecules. So to the extent you can broadly address that as well in this very thoughtful call, that'd be really helpful for us.
Okay. We think the pathophysiology of IPC and CpcPH is similar no matter the cause of the left heart failure, whether it's HFpEF, where you have a thick, stiff left ventricle that doesn't relax adequately during diastole and therefore doesn't adequately fill with blood, or HFrEF, where it's often in patients who have had heart attacks and therefore you sort of have a weak muscle that doesn't pump adequately. Both of them end up with high pressures in the left side of the heart that backflow into the pulmonary circulation. So we believe the effects on the pulmonary hemodynamics should be similar. In HFpEF, we think but we can't prove it. We think what drove the increase in cardiac output and the decrease in wedge pressure that we saw was mainly a lusitropic effect, an active relaxation of the myocardium during diastole that enables the left heart to better fill with blood. So when that PVR goes down and you can send more blood into the left side of the heart, the left side of the heart can actually accept that blood and then pump it out. In HFrEF, we think the main thing that will increase cardiac output is the fact that you dilate the systemic circulation, you decrease the resistance in the systemic circulation, and it'll be easier for a weak heart to pump blood out because it's pumping out against less resistance. That latter might also be important in HFpEF. We're just not sure it's quite as important. So I think you'll get an increase in cardiac output and a decrease in wedge pressure in the HFrEF patients as well. In fact, there were studies, right heart cath studies done with serelaxin in the past in a HFrEF population that have demonstrated that. I can't predict the degree of the magnitude of the effect that we'll see. Your second question was about the editorial. It was an editorial that was published about a month ago. The first author is the CEO of a company with a short-acting relaxin, and as you said, proposed multiple hypotheses why the efficacy or safety of a short-acting relaxin might be better than a long-acting relaxin. There wasn't a lot of evidence supporting the claims, so I'll just say a few things at a very high level. Our long-acting relaxin, TX45, signals exactly the same way as native relaxin in cell cultures experiments with cells that express the RXFP1 receptor. There's really no difference between native relaxin and our long-acting relaxin. They raised a question of immunogenicity and whether the FC could elicit an immune response. We've made mutations to RFC like others have so that you don't get that. And to date, and as far as we know, other long-acting relaxins have not been associated with immunogenicity. Obviously, we'll continue to look at that in our program. There was a claim, we've never observed native relaxin or TX45 translocating from the cell surface to the nucleus in cell culture experience. The ability of native relaxin to translocate to the nucleus and activate the glucocorticoid receptor has only been observed in one lab in 2004. And I have to tell you, I might have concerns about, I don't believe relaxin activates the glucocorticoid receptor, and I'm not sure it would be a good thing for patients if it did. And finally, there were questions raised about effects on blood pressure with a long-acting relaxin, and we have not observed any safety issues with TX45. And Lilly has put out a public statement that they didn't see a safety issue with their long-acting relaxin either.
Great. Thank you so much, team, and congrats again to the great data. I'll jump back in the queue.
Thank you. Our next question comes from the line of Tiago Fauth with Wells Fargo. Please proceed with your questions.
Great. Thanks for taking the question, and congrats on the data from me as well. Again, there's not a whole lot of comparable data across the board. You have the healthy volunteer renal blood flow for Lilly, some cardiac output for Astra, to relaxing acute benefit in a different patient population. But trying to piece everything together, how you guys feel still about TX45 differentiation across the class. And then a bigger picture question here is, again, we're starting to see emerging evidence of benefit in a few different subpopulations. Strategically, what are some of the puts and takes here? Like CpcPH is a little bit more PAH-like, right? So a little smaller, maybe you can move the needle a little bit more, higher price point versus a broader patient population. Like what are some of the levers here that you're thinking about strategically in terms of trial size, size of the commercial opportunity, so on and so forth? Curious how the data updated those thoughts. Thank you.
Okay. Tiago, remind me the first question. Okay, you wanted to know how we differentiate.
Yeah, just differentiation.
The differentiation. At this point, our base case is that our major differentiation will be on ease of administration. We have the longest half-life in normal healthy volunteers compared to the AZ and the Lilly. We think that came from extensive protein engineering that we did in multiple things, but including decreasing the isoelectric point. And we really think, based on the data that we have, that monthly dosing is feasible. We are testing an every other week regimen, but based on the data, we think there's really the possibility that monthly dosing will provide the same efficacy as every other week dosing, and that would differentiate us from both the AZ and the Lilly compound. I can make theoretical arguments based on the work we did to lower the isoelectric point, which we think could enable our drug to get better tissue penetration, that maybe with long-term dosing you'd get better antifibrotic effects. But that is completely theoretical, and I don't have data to support that, so that is not part of my base case. In terms of how we're thinking about levers, I can tell you that CpcPH is a very high unmet need population. The 5-year mortality is quite high. In some publications suggest it's even worse than PAH. Population size, as we said, depending on how you define it, PVR greater than 2 or PVR greater than 3, is 400 to 700,000 patients in the U.S. We've always said one of the reasons, a positive in going into CpcPH or HFpEF early on is we thought, as opposed to going into HFpEF, you could lock in premium pricing and access because this is the highest risk patient population with no approved therapies. So I think that there are levers, no matter if we're in CpcPH or the broader group 2 population. I think the commercial opportunity for this drug is quite high. And then, I'll tell you, you look at this data and you say, can you go into other forms of pulmonary hypertension, group 1 or group 3? I think there are multiple different ways we could go from here.
Fair enough. Perfect. Thank you so much again, and congrats.
Thank you. And there are no further questions at this time, and this also concludes today's conference. You may disconnect your lines at this time. We thank you for your participation.